Hi there. So we have one more presentation for the theater this afternoon, and it's being made by Michelle Zen, the Chief Operating Officer of Maplex. And the title of her presentation is the development of a next generation ADC technology. So with that, Michelle.
Thank you. Thank you. Well, I was given the opportunity to talk at the last minute, and it used to be my BD VP say supposed to present this, but he said today, okay, Michelle, you're here, you have to present it. Okay, so I was thrown in the box, so I will do that to my best. How many of you know ADC? I know that not too many audience already, but anybody know ADC? Yeah. So antibody conjugate, antibody drug conjugate. So Maplex, how do I do that? Hey, here you go. Maplex is basically it's a complete solution, one-stop shop for gene clone to ADC fill finish. So based in China. So we will have a process development facility in San Diego, and we have a process development facility in Shanghai, China. So, but the main facility is in Yantai, in Shandong, and China. So we basically, as long as you have a gene sequence, we can make the naked antibody, and we can synthesize the linker and the payload cytotoxin, and we conjugate them together and make the fill finish products for your clinical trial for whatever the purpose you are using. So we're a fully GMP compliance facility in China. So Maplex ADC development, we can basically doing all kind of a combination of molecular antibody, conjugation type, a linker, and a cytotoxin. Okay, so we can do all kinds of experiment for you, development for you, and then you can find out which one is the best for your drug, for your purpose. So we do traditional conjugation, okay, which is traditional cysteine conjugation. We also do traditional lysine conjugation and enzymatic method, and we also do whatever you ask us to do. But our specialty is really our, as you know, and all those technology have drawbacks, right? So antibody drug conjugate have a drawbacks. So now we have basically our own. This is whatever the traditional conjugation. You can get kind of distribute for DAR from 2 to 8, right? A kind of a polydispersity things. But our conjugation is a thiocovalent conjugation. So we covalently conjugate it to the cysteine and in the disulfide bond, and then we fix it to mainly is DAR4, right? So therefore, if you look at the picture on this side, it's like a traditional thioconjugation. You get a distribution from the ratio of a drug and the cytotoxin from 0 to 8, but in our, so we can get the ratio mostly 4. So it's more stable, consistent, and quality is very easy to be controlled. So our DAR4 ratio is like actually more than 85%. So as you can see, the stability of thiocovalent is very high for the thioconjugation. And the dosing and the efficacy is dramatically increased. And then our conjugation method was worldwide IP. So our ADC capability, we make, we are the only one can do everything in the one shot. So we do sequence to the molecular antibody or bispecific antibodies in-house GMP. We do the synthesis of cytotoxin. We do the synthesis of linker, and we do all the conjugation all under GMP. So our ADC capacity right now can up to 150 liter we have facility going up to 500 liter for ADC under GMP. And this is our ADC GMP manufacturing, okay? So showing that GMP manufacturing is in China. And then we have project, we delivered over 90 ADC projects so far for our customers. And the most advanced one is actually in the phase 3 clinical trial. And then in summary, so Maplex has a extensive capability in multiple conjugation technology. In our proprietary thiocovalent conjugation is basically our advanced technology for ADC conjugation. We have created a scale-up next generation technology that will eliminate the heterogeneity without the cost, the royalty, and the time issue of site-direct mutagenesis. And then we provide, we can, as long as you have a sequence, and then we can make it to IND batches. So most of our clients is doing dual filing or triple filing. We make the products that we file in Australia, we file in US, we file in China. So this is and then we can do from gene sequence to RBC within like 12 weeks. We can get it from gene sequence to RBC. And then we will make the master cell bank and a working cell bank and the GMP conditions and store the banking for you, and then make it the GMP batches from 200 liter to 2,000 liter single-use bioreactor. And we are the big customer of Sartorius. So all our equipment are Sartorius, from 2 liters process development to Amber to 500 liter, 1,000 liter, and a 2,000 single-use bioreactor. So our phase 2, like an M2 expansion, is up to 24,000. We have 12 times 2,000 single-use bioreactors. So total is 24,000 plus our M1s, which is another 2 times 2,000 liters. So our capacity is way ready for commercial manufacturing. So we can transfer your technology to us at any stage. And then we can start from sequence 2. So whatever the stage you are, and we can do it. And our executive team members, just like me, we all come from US. Myself has been in North America for more than 30 years, for close to 30 years. Have extensive experience in pharmaceutical company, small, large, biosimilar or innovator. So our executive team members all come from US. So that's what the advantage of Maplex, and we can try to bridge in 2 countries' knowledge, experience, and the cultures together, and make sure we have the first-class service to our clients. So if you want to have more information, you can come to talk to me after presentation, or visit our booth at 1701, and then we have more information available for you. Thank you for your attention. So I will be happy to answer any questions if you have. If not, and then we can, talk about it after this presentation, and you're welcome to approach me. Thank you.